Hello everyone, welcome to my channel The Tooth Fairy, and our today's topic is types of hemolysis. It is of three types: alpha, beta and gamma. In this video we will read a little about what is blood agar, composition, uses. Since I have already posted a video about culture mediums, there it is explained nicely. यहाँ पे हम थोड़ा सा ही पढ़ेंगे. The main topic is principle of hemolysis and types of hemolysis. So how the question is asked? Very rarely it is asked as a 5-mark separate question that what are the type of hemolysis, very rarely. But frequently it is asked for vivas. They will show you a petri dish on which you can easily identify that it is blood agar. Okay, but the problem lies that which bacteria it is. So based on the hemolysis it is showing on blood agar, you can easily identify which bacteria it is. And then sometimes they ask about the composition of blood agar also. And other than that, in bacteriology you will in each chapter almost you will read about that it is showing alpha hemolysis, it is showing beta hemolysis, so you should know about it. So blood agar, why we use blood agar? Since some bacteria have special demands, okay, they cannot grow just here and there, so those bacteria are called as fastidious bacteria. यहाँ पे fast word आ रहा है, so like we humans keep fast, and then we have special demand that we have separate chips, separate cereal, so we have special demands when we are fasting, same as bacteria. Then this is how it looks, reddish color. The McConkey agar looks pinkish, and the chocolate agar is basically heated blood agar, which looks brown in color. So composition of blood agar, it's just nutrient agar plus the 5% sheep blood. And why sheep blood we are using? Because human blood has some inhibitors and it will not allow the bacteria to grow on it. So we are using sheep blood. The pH should be slightly alkaline, that is 7.2 to 7.6 range. Then its uses. First uses that we are using it for growing pathogens. Hemophilus influenzae and Neisseria species, they grow on chocolate agar, and that is heated blood agar. Then it is used to detect and differentiate hemolytic bacteria. Depending upon which type of hemolysis it is causing, we can differentiate the bacteria. Now, principle of hemolysis is that based on which enzyme is present in bacteria, every bacteria break down RBCs in different ways and to different degrees. So this helps in distinguishing different bacteria. Now the types of hemolysis, there are three types: alpha, beta and gamma. The other one we can read about is alpha prime or wide zone alpha hemolysis and the target hemolysis. That one is different. So last के जो ये दो हैं, that is for just additional knowledge. The target hemolysis is given in your book. Now how we see hemolysis, तो अगर पेट्री डिश है उसको हाथ में नहीं पकड़ के रखना या उसके ऊपर टॉर्च मार के नहीं देखना कौन सी hemolysis है, तो you will hold it against the light source and observe with the light coming from behind. Okay, that's how you will check out what type of hemolysis it is. Now if you see this picture, everything is given in this. So in alpha there is greenish discoloration, and in beta there is complete discoloration. That means you cannot see the red color here, so it is completely discolored, the medium is completely discolored. What is this? जो बीटा शेप है यही bacterial colony है. Okay, the bacteria started to grow and it formed a colony. Now these bacteria secreted few enzymes, and depending upon which enzyme they secreted, they completely or partially destroyed the RBCs. And in gamma, there is no hemolysis. So alpha hemolysis में there is partial lysis of RBCs. How it occurs that it converts iron in hemoglobin. It oxidizes iron of hemoglobin and then it converts RBC hemoglobin to meth hemoglobin. Now this meth hemoglobin gives a greenish brownish discoloration around the colony. Examples are Streptococcus pneumoniae, which causes pneumonia, and then is Streptococci viridans. Viridans का मतलब भी green होता है. This is separate streptococcal group. Then we have beta hemolysis, in which there is complete lysis of RBCs. There is complete colorless zone around the colony. सारा जो red color था blood का वो कुछ नहीं रहता and it is completely colorless. Main example हमने करा था last video में I told GAS gas, that is group A streptococci pyogenes, and then there is group B also, that is streptococcus agalacti. Streptococcus agalacti causes bovine mastitis. Mastitis is inflammation of mammary glands of the cattle. तो ये होता है. And then beta hemolysis, they show it is better when there is more carbon dioxide concentration. So we can easily observe beta hemolysis colonies, since the bacteria are able to do hemolysis better in CO2 concentration, when there is increased CO2 concentration. Then the examples are Staph aureus, ये भी हमने पढ़ लिया, and then Listeria and Bacillus. If you want to read, you can, but main examples you should read is Streptococcus pyogenes and Staphylococcus aureus. Then we have gamma or non-hemolysis. Then there is no hemolysis of RBCs. There is no change in the medium surrounding the colonies. जहाँ पे भी bacteria grow हुआ उसके आसपास के medium में कोई change नहीं आता. Example is E. faecalis, Enterococcus faecalis, and then Staphylococcus saprophyticus and Staphylococcus epidermidis. We did in last video of Staphylococcus, जो हमने बोला था coagulase negative staphylococci. Now here you have some more examples like beta hemolysis में Streptococcus pyogenes, it is completely discolored and यहाँ पे greyish color है E. coli का. And then gamma hemolysis में there is cons bacteria Staphylococcus epidermidis and Staphylococcus saprophyticus. Main आपको याद रखने हैं E. coli, Streptococcus pyogenes, Staphylococcus, and Streptococcus pneumoniae and Streptococcus viridans. The next one is alpha prime or wide zone alpha hemolysis. Okay, so यहाँ पे this is not included in your book. It is just for your general knowledge if you want to add something extra in your brain. So here you can see that this is bacterial colony and then there is medium blood agar tha which is no change, अभी भी red color का है, ठीक है. So that means colony के बिल्कुल पास there is small narrow zone of intact erythrocyte, but then there is a very wide zone of complete red cell hemolysis, ठीक है. तो कई बार by mistake they will say कि ये beta hemolysis है because they ignored the narrow zone of intact erythrocyte. Okay, and then this is confused with beta hemolysis. Most reliable method to appreciate this narrow zone of intact erythrocyte is using rabbit blood in heart infusion agar. So instead of using sheep blood agar, we will use rabbit blood in heart infusion agar. It is shown by some strains of Streptococcus causing mastitis in bovine mastitis, that is inflammation of mammary gland in cattle. Then this is given in your book. This is target hemolysis. So target is how it looks like. And then इसका example is Clostridium perfringens. Clostridium perfringens gives characteristic double zone hemolysis that is shown as target hemolysis. Now what happens is उसमें जो था पुराने वाले में there was intact zone, ठीक है. लेकिन इसमें क्या होता है, there is narrow zone of complete hemolysis which is caused by theta toxin, and then there is wider zone of incomplete hemolysis that is due to alpha toxin. So Clostridium perfringens में theta toxin की वजह से there is complete hemolysis but there is very narrow zone and a wider zone due to alpha toxin. तो यहाँ पे अगर हम we can easily appreciate that this main thing is here is colony. Then there is complete discoloration which is due to theta toxin, and then there is partial discoloration and it is wider zone which is due to alpha toxin. So one important thing here is Clostridium perfringens also causes food poisoning. In the last video we read that Staph aureus also causes food poisoning, but उसके symptoms appear after 2 to 6 hours. Clostridium perfringens में it appears 8 to 12 hours, and it is mainly because of some cooked meat item or some poultry item like eggs. And then there is other one, Clostridium botulinum. So Clostridium botulinum का होता है 12 to 24 hours के बाद, and it is mainly because of some canned food, okay, some preserved food. So and that is very deadly, that Clostridium botulinum. Botulinum toxin की वजह से होता है उसमें, and it is one of the most toxic toxin. So यहाँ से आपको पता चलेगा कि अगर फूड पॉइजनिंग हो रही है तो क्या आंसर है उसका, कौन सा बैक्टीरिया कर रहा है, क्योंकि तीनों ही कर रहे हैं, बट कौन सा कर रहा है डिपेंड्स अपॉन क्या खा के हुई है एंड कितने घंटे बाद हुई है. Okay, thanks. I hope this video was helpful. Please like, share and subscribe.